Thanks, Alexey.
All right. Thanks very much, Garen. Thank you very much for showing up, everyone who's here listening to me. So I will talk today, switch the gears really a lot from what we've heard before in the previous talk, and talk about the nuclear protein RCD1 in Arabidopsis. And in this first slide, you can see the nucleus and RCD1 sitting in this nucleus in this yellow bush that is, I tried to draw poly-ADP-ribose. And on top, you see the organelles, energy organelles, mitochondria, and the chloroplast. So in the first part of my talk, I will describe you briefly what we know about the connection between energy organelles and this nuclear protein RCD1. And then in the second part of this talk, we will go into the realm of mechanisms, or have a glimpse into the mechanisms of what's going on in the nucleus to integrate the signals from the organelles and from the other signaling pathways. But I will start by introducing this RCD1 knockout mutant. As you can see, it's a very unhealthy plant, and it's been isolated in our lab over 20 years ago due to its high sensitivity to ozone. And this is how it got its name, radical-induced cell death. And then over the years, several groups studying RCD1 have found a lot of different and contrasting phenotypes. It was deficient in development, in stress responses, it was tolerant to some things and sensitive to other things. So for example, being sensitive to ozone, RCD1 mutant is extremely tolerant to methylviologen. And this is what I will expand in the next few slides. So here we look at chlorophyll fluorescence of wild type col0 and RCD1 plants that have been pre-treated with either water or methylviologen. And then we turn on the light. So this is methylviologen, also known as paraquat. It's used as a herbicide. And then we turn on the light, and things start happening. Actually, when we look at chlorophyll fluorescence, we will see the gradual decay of efficiency of quantum efficiency of photosystem 2 that occurs in the wild type, but not in the RCD1 mutant. I don't know why this is not working. All right. So there was a movie that shows that this actually these plants are dying while RCD1 mutant is completely tolerant to this treatment. So what is going on? Here is a chloroplast with its two photosystems, photosystem 2 and photosystem 1. So when we turn on the light, we turn on the electron transfer pathways in the chloroplast. Here we see a linear electron flow, cyclic electron flows, electrons flowing to some electron acceptors inside chloroplasts or outside of them, and also electrons jumping onto molecular oxygen, which generates reactive oxygen species, ROS. So once we drop in methylviologen, we divert most of the electron transfer in the chloroplast to formation of reactive oxygen species. So at the expense of all the other or many other electron transfer pathways. Interestingly, the effect of methylviologen on this photosystem 1 and formation of ROS is the same in both the wild type and the knockout mutant. So we study this by spectroscopically analyzing the photosystem 1 redox state, and we see that this redox state is exactly the same with and without methylviologen in the wild type and in the mutant. So the excess of methylviologen to photosystem 1 is not the reason for the tolerance of this mutant. But then later, if we give enough time, then we see that photosystem 2 is dying in the wild type, but not in the RCD1 knockout. So we can draw this inhibitory line here, but mention that it's not happening, this inhibition is not happening in the RCD1 mutant. So RCD1 is shown here in the left panel as these purple dots inside the nucleus. We can see it in the nucleoplasm and in the nuclear bodies inside the nucleus. And chloroplasts are these green structures. This is a mesophyll cell of Arabidopsis. So the question is, what is the connection between the nuclear protein, RCD1, and the processes in the chloroplast? So we spent quite many years trying to understand what is this connection, and the first most natural experiment to do would be to look at the transcriptomics, because RCD1 is sitting in the nucleus, so we expect it to be implicated in gene expression. And here are the highlights of the gene expression changes in the RCD1 knockout mutants. So we were sort of expecting to see some chloroplastic components that could explain methylbiology intolerance. However, after thorough analysis, we actually see very few genes whose products are located to chloroplasts, and these genes cannot really explain methylbiology intolerance of the RCD1 knockout mutant. But then looking closer at the list of the genes misregulated in RCD1, we noticed that a lot of these misregulated genes have to do with mitochondrial retrograde signaling. So here I introduce another player in this scheme, the mitochondrion. It has its own electron transfer chain, going from complexes 1 and 2 through complex 3 and then respiratory complex 4. And one very widely accepted way to study mitochondria in vivo is the use of the blockers. One of these blockers is antimycin A. So antimycin A blocks electron transfer through mitochondrial complex 3 over here. And once you apply it, you see accumulation of electrons upstream from complex 3, which cannot go through. And this also initiates leakage of electrons to the outside surfaces of the respiratory complexes, which creates reactive oxygen species. These reactive oxygen species activate as a family of transcription factors. And these transcription factors migrate into the nucleus, and they induce expression of the so-called MDS, mitochondrial dysfunction stimulon. This is a set of genes that is responsive to the mitochondrial retrograde signal. A subset of these genes includes mitochondrial alternative oxidases. So these alternative oxidases accumulate in the mitochondrion, and they produce an alternative respiratory pathway that actually helps to bypass this traffic jam of complex 3. So this is the regulatory loop that I just described, having to do with mitochondrial retrograde signaling. So the next question is, what does RCD1 have to do with this regulatory loop? So we see biochemically that RCD1 is binding ANAC transcription factors. We see this in vitro. So this is the NMR scatter that shows RCD1 with and without ANAC peptides. And we also see this in vivo, for example, here, actually in a heterologous expression system in the HEC cells. Also, RCD1 binds ANAC transcription factors that are involved in mitochondrial retrograde signal. And also, we have done a lot of experiments for which you can have a look at our publication in eLife from 2019, which show that RCD1 is actually inhibiting the function of ANAC transcription factors inside the nucleus. You can view it here. Basically, you have this yellow arrow. So if you think of an analogy from electronics, you could imagine it looking this way. So you have an input of the signal from the organelles, and then the output that goes to gene expression. And RCD1 is the gateway that is controlling this circuitry over here. So when RCD1 is missing, then this signal is constantly induced. So ANAC transcription factors are constitutively active. MDS genes are always induced in standard growth conditions. Alternative oxidases start accumulating, and this we see very well biochemically. RCD1 mutant accumulates 10 to 15-fold more alternative oxidases than the wild type col0. And this is accompanied by very significant changes in all different parts of metabolism, including, for example, respiration. So here's just to illustrate what sorts of defects in metabolism we see in this mutant, is to show you that this mutant is tolerant to cyanide. So cyanide blocks electron transfer through complex 4. And when you apply it, then seedlings stop respiring, in the wild type seedlings. They stop consuming oxygen, which you see here. And RCD1 is still very tolerant to cyanide because of the activity of alternative oxidases. So we have done a lot of metabolomics analysis on this mutant, and we actually see that not only respiration, but the cell wall processes, the starch, and the different, carbohydrate metabolism and many other aspects are very much distorted in the RCD1 mutant. Among others, we see that these alternative oxidases also contribute to the processes in the chloroplast. For instance, when we apply the inhibitor of alternative oxidases, sham, we see that the tolerance of RCD1 plants to methylviologen go down. So it looks like AOXs are part of the story why the RCD1 mutant is so tolerant to methylviologen. We also see some effects on chloroplast linear electron transfer, which suggests that there is a pathway that connects electron transfer pathways in the chloroplast and activity of alternative oxidases. We are now actively studying what are exactly the mechanisms of this pathway that connects the two organelles. And last but not least, for this first part of the talk, is that when we apply methylviologen to generate reactive oxygen species in the chloroplast, or when we apply hydrogen peroxide just to have a boost of reactive oxygen species in the plant tissues, we see that the levels of RCD1 go down. Moreover, if you separate this protein extract on the non-reducing gel, you will see that already after 30 minutes of this ROS-inducing treatment, you see that RCD1 gets assembled into oligomers that are bound by thiol bridges, and we think that these oligomers are actually some kind of inactivated forms of RCD1. So altogether, we draw another arrow here that shows that reactive oxygen species from the chloroplast are inhibiting RCD1, thereby affecting this whole regulation that I described before. So this is my first summary. RCD1 inhibits ANAC transcription factors that mediate mitochondrial signaling. In the mutant deficient in RCD1, this mitochondrial signaling is constantly induced. This leads to accumulation of alternative oxidases in the mitochondria, which affects respiration and metabolism. This also affects the chloroplasts. And the reactive oxygen species produced in the chloroplast are dictating the stability and oligomerization of RCD1, thereby regulating this loop. Right, so now we come to the second part of my talk, which is the nuclear, what's going on in the nucleus. So we see RCD1, and I will start by showing you the domain structure of this protein. So everything I told you before was related to this C-terminal domain of RCD1 that is called the RST domain. So we solved the structure of this RST domain, and this is the domain that is actually interacting with transcription factors. And these are not only the ANACs. Our colleagues in the University of Copenhagen have studied this RST domain in detail, and they show that this is a universal transcription factor trap that can capture transcription factors of different classes. And indeed, in the yeast to hybrid assays, we see that RCD1 binds not only these ANAC transcription factors that I described to you, but also transcription factors implicated in the heat stress, in salt stress, in light responses, in development, hormonal signaling, and so forth. This makes this RCD1 protein a versatile component of regulating many different signaling pathways. So what about the first two domains of RCD1? So the first one is called WWE for the amino acids that are very conserved in this domain. And the second domain is called PARP-like, poly-ADP-ribose polymerase-like. So maybe not all the plant scientists have been frequently hearing about poly-ADP-ribose. So in the next couple of slides, I will show you what this is. So studies of poly-ADP-ribose are really explosively developing in mammals now. So this is the electron carrier and the NAD+, and this part of it, of the molecule, is called ADP-ribose. So the enzyme called poly-ADP-ribose polymerase can connect covalently ADP-ribose to the acceptor protein. Moreover, it can connect another unit and another unit and another unit, making a long polymer. And this polymer can also be branched. And this polymer is called poly-ADP-ribose, or PAR. So PAR has been initially described as the component that is involved in DNA damage responses, but then recent studies show that it's also involved in as a scaffold molecule, and also that it can contribute to the liquid-liquid phase separations. It is implicated in signaling and many other processes, and most of them are studied well in mammals and plants. The PAR metabolism and functions are not that well understood yet. So you can think of the PARP as the enzyme that is adding PAR to the acceptor proteins. Then PAR glycohydrolases are removing this modification from the proteins. And then there are the PAR binding proteins that bind non-covalently to this PAR polymer. The PARPs can be called writers, PARGs can be called erasers, and PAR binding proteins readers. This is the set of all proteins in Arabidopsis that contain this PARP domain. So on the top, there are these enzymes that are active PAR polymerases. And here on the bottom, we see the proteins that also have the PARP-like domain, but they are lacking the three crucial amino acid residues in the active center. So they are considered to be inactive PAR polymerases. Just for a comparison, have a look at the Homo sapiens set of PAR proteins. We again see similar patterns. We have these PARP domains, sometimes accompanied by the WWE domain, like in RCD1 here. What we never see in humans is this yellow RST domain at the end, because that is plant specific. So our research suggests that the WWE and the PARP-like domains actually form a unit to bind poly-ADP-ribose. And we see this effect in vitro. We see that this is the surface plasmon resonance study that shows that RCD1 is binding poly-ADP-ribose, and if you mutate away the WWE domain, for example, you stop having this binding in vitro. But also you see the evidence of this happening in vivo. So here you see the nucleus and RCD1 localized to the nucleus, to nucleoplasm and nuclear bodies. And when we treat these plants, these leaves, with PARP inhibitor that inhibits formation of PARP, the nuclear bodies go away, which suggests that maybe PARP is the essential components for these nuclear bodies to form in vivo. Also, when we mutate away the WWE or the PARP domain, but not the RST domain, we stop seeing RCD1 localized to the nuclear bodies. So altogether, it suggests that these two domains are involved in binding to PAR, and that this binding helps the protein to localize to the nuclear bodies. In these nuclear bodies, RCD1 meets partners, and we are now actively studying this, the interactome of RCD1 and the nuclear bodies. And one of the sets of proteins that it meets in the nuclear bodies are the phosphoregulatory protein kinases, the PPKs, sorry, photoregulatory protein kinases. So here in this image, you see RCD1 that is forming nuclear bodies, PPKs forming nuclear bodies, and the very nicely merged signal from both channels. These PPKs have been studied and known as regulators of photoreceptor signaling, of abscisic acid signaling. They are binding to components of phytochrome or cryptochrome signaling and phosphorylate them, and this phosphorylation targets them to degradation frequently. And actually, we see that if we mutate away the phosphosites that are phosphorylated by these PPK kinases in RCD1, then RCD1 starts accumulating to higher amounts than the wild-type RCD1. So it looks like this binding to PPKs in the nuclear bodies is facilitating degradation of the protein. Here is the slide of the pictures, the photos of these of the different complemented lines of RCD1. So on the very left, you see the wild-type plant. Next comes the knockout mutant. Next comes the mutant complemented with a wild-type version of the gene. And then on the right, you see the the version that is complemented with a RCD1 that lacks the RST domain. So if you don't have RST domain, you cannot trap all these different transcription factors, and you get a beast that looks very different both from the wild type plant and from the RCD1 knockout mutant. It's just a totally different set of phenotypes that we observe. And yet another type of phenotypes we observe if we knock out the WWE domain or the PARP domain. Then we have a yet another looking plant that has different tolerance to many agents. It has different flowering time, developmental, and other phenotypes. So I show you this slide too. I think this is really a miraculous illustration of how these different domains contribute to changing the habitus, the stress tolerance, and the development of a plant. Which brings me to the one but last slide, where I developed that idea of electronics. So I think, and I also took the liberty of instead of writing RCD1 to say SRO proteins in general. So we think that inside the nucleus, we have signal inputs from many different transcription factors and many different signaling pathways, including organelles, photoreceptor, and so forth. And SRO proteins act as signal mixers that fine-tune the signal from each of these inputs to generate the consensus output on gene expression. So RCD1 is a novel plant reader. It binds many transcription factors. It's localized to nuclear bodies in a PAR-dependent way. It is extensively regulated at a post-translational level. You have seen that it's been regulated redox by through thiol redox regulation by the ROS produced in the chloroplast. You've seen it's also regulated by phosphorylation through PPK kinases. So thereby RCD1 acts as a signal mixer of many signaling pathways emitted by the energy organelles, photoreceptors, and so on. And the still remaining mystery is what actually are the components, or what are the proteins that are PAR-related, that are through the PAR, the binding partners of SROs. So that's my talk, and I would like to thank all the people who have been involved in this. We had a lot of collaborators over the years in different universities, and here is our core group. Jaakko Kangasjärvi, Julia Vaininen, Julia Krasensky-Vracek, Richard Gossens, Michi Vracek, and Mikhail Brochet. So this is our core group of people who are working or have been working in the University of Helsinki on RCD1, and we are funded by Finnish Academy of Sciences. Thank you very much for your attention.
Thank you very much, Alexey. So interesting talk. Certainly a lot going on there with RCD1, and still a lot to find out for sure. So once again, I encourage people to ask questions. I have a few to kick off, one or two. So you mentioned that it's a plant-specific protein. So I guess the mix of the domains is a plant-specific protein. Is it across all plants, or just in higher plants, or what have you looked to see that?
It's present in the higher plants. It's present in the higher plants. There is a specific, so as you may have noticed, it has a sibling in Arabidopsis. It's called SRO1, similar to RCD1. Okay. And the mutant deficient in that gene has virtually no phenotypes. So that split between RCD1 and SRO1 homologs, which is quite mysterious, and we still don't quite understand the reasons for such a diversity of functions, is most pronounced in Brassicaceae.
Okay.
While in the other species of higher plants, we don't really see that split that well. But yes, I mean, it is higher plant specific.
Okay, but all plants have, mitochondria and chloroplasts. So the link between those would be, you would predict, be facilitated by another protein playing the same role as RTC1, or do you think there's a higher plant specific function?
To be honest, so you mean about algae, for example, right?
Yeah, for example.
To be honest, I cannot really answer this question now. I think that in the higher plants, we definitely expect to see some sort of regulation like this. I would refer you to our publication from 2016. There was a link in my talk, which describes that the phylogenetic analysis of these SRO proteins across the green cut.
Okay.
So function wise, I would suspect that they could act similarly. Yes, as well in ferns and in bryophytes. Yes.
Okay, so let me ask just a final question for myself. So the pictures of the whole plant phenotypes there you showed were Arabidopsis plants grown in short days. So do you see similar phenotypes in long day plants, or?
So this was 12-hour day, which is kind of in between, right?
Okay.
So yes, we do see these phenotypes across different day lengths.
Okay.
Also the flowering phenotypes. Yep.
Okay. Okay. So a question from Dimitra Angelopoulou. Sorry if I pronounced the name wrong there, but they ask, have you done any work with plant pathogens in these mutants, or do you know if RCD1 is involved in hormone signaling at all? So pretty broad.
Yes. For this, I would refer you to publications on RCD1. There's been quite substantial work done on the pathogen resistance. So briefly, RCD1 is quite sensitive to pathogens. It's very tolerant to the reactive oxygen species related processes in the organelles, at the same time being very sensitive to the pathogens. And the reasons for this interesting contrast are not yet clear. But yes, there were the studies of RCD1 resistance to pathogens as well. And also about the hormones. So we had a recent publication where we see the connection of RCD1 phenotypes to hormonal signaling.
Which hormones specifically?
So we looked at ethylene. We looked at salicylic acid is another hormone that in relation to which RCD1 mutant has a phenotype.
Okay. And that could be defense related, I guess. That is
defense related. Also in gene expression, we see the changes in the actually down-regulated genes that are involved in the pathogen responses.
Okay.
So that could be one of the reasons of the sensitivity of this mutant to pathogens.
Okay. So I would encourage anyone else to ask a question, but let me continue. So I was intrigued by those nuclear bodies, actually, so really interesting. And also then trying to link that with the fact that when you get the ROS burst, you get the large increase in the high molecular weight complexes. So are those two things linked together possibly? So if you ROS burst, you get more nuclear bodies formed?
Right, that's a very good question. We do see the changes. So when we have this chloroplast production of ROS by using methylbiologen, we do see in subtraction of nuclei the changed localization of RCD1. So the patterns of these nuclear bodies change. But this is not happening in 100% of the cells, so it is a difficult subject to quantify.
Okay.
The short answer is yes, we do see some effects.
Okay. So question from Paige Panther, and she asks, do you know if there's any circadian regulation of RCD1?
Yes, there is. Actually, now we are also trying to develop this connection of RCD1 to circadian regulations, because we know that PAR is also implicated in circadian rhythms. And I cannot now tell you about the gene expression of RCD1 gene, but we know that stability of RCD1 protein is changing throughout the day.
Okay, cool. Okay, so I guess we'll leave it there. So if you have any other questions for Alexey, I'm sure he wouldn't mind answering them. If you'd write him an email, no problem. But thanks to everyone who's attended today. And as I